Hi, good evening, everyone. So today we will do discussion for our case study. There are total of 4 case study together. My part is case study 1. So my name is Nurul Aimanafka binti Nur Azruddin, and my matrix number is 210213, together with my teammates Sabrina, Ain, Farisa, and Zulmaki. So I will start now my part for the case study 1 explanation. So together, there are 3 types of protein: protein A, protein B, protein C. So for the first one, protein A, I will explain to you all of the signs, symptoms that this protein has caused. It is caused by the bacteria called Klebsiella pneumonia. So the symptoms that it might show is bacteria can cause infection in different parts of the body, such as at the lungs, bladder, brain, liver, and so on. But the most common infection is a urinary tract infection. When it infects the lungs, it can cause pneumonia, which is characterized by fever, chills, cough, chest pain, shortness of breath, and mucus that may be thick, bloody, or have a current jelly appearance. Other symptoms include nausea and vomiting. So all the signs that I have told you, it matches what the diet patient go through. Next, moving on to the next protein, which is protein B. It is caused by Burkholderia cetulosapatia J2315. So the signs or symptoms include it leads to cystic fibrosis that can cause thick, sticky mucus to build up in the lungs, digestive tract, and other areas of the body. The symptoms that these bacteria can include fever, cough, congestion, shortness of breath, and wheezing. But to highlight, nausea and vomiting are not common symptoms of this infection. So this infection partially matches what the patient go through, which it can cause shortness of breath and include fever, cough, but it does not go through nausea and vomiting. So moving on to the last protein, which is protein C, it is caused by Bacillus anthracis. Based on my findings, Bacillus anthracis can cause 3 types of anthrax. So for the first one, it can cause cutaneous anthrax, next gastrointestinal anthrax, and then lastly is inhalation anthrax. So the symptoms include fever, but it is rarely present for the cutaneous anthrax. It is caused by patient who had contacts with animals. And then for the gastrointestinal anthrax, it is caused by abdominal anthrax. Symptoms are like nausea, vomiting, anorexia, fever. And then lastly, for the inhalation anthrax, the illness begins insidiously with flu-like symptoms of mild fever, fatigue, non-productive cough, and then it usually takes 2 to 5 days after initial exposure. So what I'm trying to say is this protein does not match all the signs that the patient go through. It does not match at all because the symptoms is very contradicting than the detail that the patient go through. So to conclude, the protein that matches all the symptoms that patient go through is for protein A, which is caused by Klebsiella pneumonia because it matches all the symptoms, which is fever, and then it can cause nausea and vomiting, and then there's also thick mucus. So I will choose protein A as the cause of death of the patient. That is the end of my findings for case study. I will pass to the next case study.
So now I will present about case study 2. So the pathogen associated with the information provided by Mr. Rodney is Klebsiella pneumonia. While he suspected K. pneumonia to be the cause of his death, our team believed otherwise. Upon using the pattern as a query in Fiblas, K. pneumonia did not appear at the beginning of the result list, indicating a lesser relevant to the query. This bacterium typically colonize human mucosal surface in the oropharynx and gastrointestinal tract. An infected individual may experience symptoms such as cough, fever, pleuritic chest pain, and shortness of breath. However, Mr. Rodney also exhibits vomiting, which is not a symptom typically associated with the pneumonia infection. Next, in the result list of fibroblasts, the first sequence that matched the query pattern was Bacillus cereus. This indicates that Bacillus cereus is the most similar to the query and share the highest percentage of the identity. Bacillus cereus is a gram-positive bacteria that can produce toxin and commonly found in soil, vegetation, and food. It often causes intestinal illness characterized by nausea, vomiting, and diarrhea. The infection is unrelated to the respiratory tract, thus it does not cause shortness of breath. Additionally, Bacillus cereus is transmitted through the ingestion of contaminated food, making it less likely that Mr. Rodney ingests the white powder if it was contaminated with Bacillus cereus. Another potential pathogen is Clostridium difficile, which match the query pattern. Although this sequence was not found at the beginning of the result, the possibility of the C. difficile was causing Mr. Rodney death should be considered. C. difficile is a gram-positive, spore-forming bacterium that produces toxin. The primary mode of transmission is through the fecal oral route, leading to the colonization primarily in the last intestine. So the symptom of the C. difficile infection include watery diarrhea with the mucus or occult anorexia, nausea, vomiting, low-grade fever, and lower abdominal pain. However, C. difficile infection does not cause shortness of breath and is typically not severe enough to result in death. So lastly, Bacillus, our most likely pathogen that cause Mr. Rodney death is Bacillus anthracis. Bacillus anthracis is a gram-positive root-shaped bacterium that causes anthrax. Anthrax is a deadly disease that primarily affects livestock but can also affect humans. Illness from anthrax can occur through inhalation. An infected individual develops symptoms such as shortness of breath, vomiting, and fever within 2 days. This infection can spread, affecting a gastrointestinal tract and causing body vomiting. This aligns with Mr. Rodney's observation of blood on the paper he wrote before his death. That usually occur within 72 hours, and Mr. Rodney passed away after 2 days. Additionally, it is a significant piece of evidence is white suspicious powder. In 2001, anthrax spores were deliberately placed in the powder form and used as a biological agent in male later to cause harm. It is possible that the white powder Mr. Rodney encountered was contaminated with microscopic spore of Bacillus anthracis, leading to his infection and subsequent demise. So, in conclusion, from case study, conclude that Bacillus anthracis was the causative agent of anthrax and responsible for Mr. Rodney death. It is highly likely that he suffered from the inhalation anthrax due to the presence of microscopic spore in the white powder. Bacillus anthracis matched one of the sequence that corresponded to the exact pattern left by Mr. Rodney, and his symptom aligned with those of an anthrax infection.
Okay, so I'll do a bit conclusion about this case study too. So through the PHI blast, fibroblast search, and further investigation, we have made progress in identifying the causative agent. So the fibroblast search revealed 4 proteins with similarity to the modified query sequence. These proteins are associated with specific pathogens that could be responsible for the infection. Bacillus anthracis, like what Ain said before, emerged as a strong candidate based on the symptom and potential spore inhalation. It causes anthrax, severe infection, like flu, and followed by respiratory distress. To confirm the identification, we conducted genetic sequencing and phylogenetic analysis to compare and obtain sequence with known pathogens. Through sequence comparison and functional analysis, we aim to pinpoint 2 exact pathogens responsible for the infection, and the next step involved in further analyzing the proteins and their associated pathogens study virulence factor, drug resistance, and treatment option. In conclusion, our analysis using fibroblasts and subsequent investigation provide a valuable insight to the potential pathogens responsible for the infection, while Bacillus anthracis remains as a strong candidate, and confirmatory investigation, including genetic sequencing and phylogenetic analysis, are essential to reach a conclusive identification. So we both agree that the Bacillus anthracis is our result for this case study. Okay, I'll pass to the next.
Hi, I'm Zul Hakim. So basically, I will make it very, very short for the case report for case study 3. So for the case study 3, I need to use to find the pathogen using the SciBlast method. So in order to do the SciBlast methods, for the last iteration, I will find most of the sequence are coming from Bacillus series. I will go to this straight to the point. You can say the casual pathogen is Bacillus series, and it can also cause inhalation extract since it quite similar to the symptom that Mr. Rudney has felt before, which is shortness of breath, vomiting, and high fever before he dies. To top it all, we were able to correctly identify the protein sequence as a domain rather than a toxin using the SciBlast methods which I mentioned before. The set domain containing protein lysine and methyltransferase is the protein most likely to associated with the given sequence, according to our team's thorough analysis. Furthermore, given its known trace and Mr. Rudney symptom, which is the symptoms I mentioned as shortness of breath, vomiting, and high fever before he died, which included shortness of breath, nausea, and high fever. Bacillus series is a pathogen that may be to blame for the infections. Like other kind of symptom that Bacillus cereus may bring is like inhalation attracts and also lung infections. Because of the finding from SciBlast that the protein is mostly homologous to the Bacillus genera studied by the Mr. Rudney's co-worker, with whom he had recently argued. So basically, from back to the topics, basically Mr. Rudney may have been the victims of a murder. So that's just all for case study 3. So I will pass to the case study 4.
Alright, for case study 4, I would like to explain in short where at first the death is caused by the acute lung poisoning, which caused the patient to suffer from chills and shortness of breath, nausea, and vomiting for 2 days before being admitted. So he was also said to mutter the word 6 of 6 before dying. So there was an extraordinary inflammation of the lungs and occurrence of a yellow mucosal membrane. The mucus is isolated and the unique protein is sequenced. So next. First, we analyze the information according to the given instruction. So since we get the protein sequence, we compare the protein query sequence against a nucleotide database. So we choose 6 different species from the results and use MEGA to produce the phylogenetic tree by maximum likelihood method. So from the phylogenetic tree shown, we have concluded that the given sequence in the case study for almost identical to Dichomimus squalens. So due to certain reasons also, we also use the protein sequence given straight away instead of using BLASTP to search for the identical genomes. So BLASTP is used to detect comparable or homologous protein sequence. A protein query sequence is compared to a protein database. So we get 6 sequences from the result and use neighbor joining method to create the phylogenetic tree. So from the phylogenetic tree produced, we can conclude that Bacillus thuringiensis is being same as the sequence that given in the case study 4. So the first result, which is Dichomotus squalens, is matched with the sequence given. However, the organism that we make a research on it doesn't cause any health issues to human because it is a wood decaying fungus. So at the same time, we also doesn't found any articles that cause respiratory problems or any health effect to humans. So next is Bacillus thuringiensis. So this Bacillus is the organism also which match to our sequence. So to compare the case report 4 sequence, we made a pairwise alignment from the sequence given with the Bacillus thuringiensis. So we found that it is 100% identical to each other. So we also made a research on the Bacillus thuringiensis, which it will cause some health issues to human. There is an article also provide that information regarding to Bacillus thuringiensis cause pulmonary infections in humans. Thus, we have concluded that the pathogen cause problem in our case report 4 is Bacillus thuringiensis. So next, you guys think that what pathogens that really cause the disease?
I still think that it was a Bacillus anthracis that caused the disease. What about you, Farisa?
Yes, I agree with Ain as I have convinced to all of you guys just now. Like even though we do the same case study, we can see like the most related is Bacillus anthracis, where it relates to the inhalation and the symptoms given in the case. Does anyone to continue?
I also agree with Ain and Farisa because from what they have presented, I also feel that those symptoms are the most I think it matches all of the signs that the problem has caused. So I agree with them.
I also agree with Nur Ain because it's simple. It's very, very simple because logically, Bacillus anthracis is the answer because it's like overlapping in most of the cases. That just makes sense to me.
Okay, so we have found that using different methods and then we have the different given sequence of the pathogen that is given. So we can found that majority agree that Bacillus anthracis is the pathogen that caused the people to death. So anyone that want to have a different ideas about what pathogens that will cause again? No.
It's like I just want to add something. It's not summarized, but I just summarize lah. So I think most of us agree that it's Bacillus anthracis because, like I said before, it's overlapping between most of the cases. So although Bacillus cereus and Bacillus anthracis can cause pneumonia-like symptoms, these suspects, Bacillus anthracis is the casual pathogen of Mr. Rooney's death because Bacillus cereus rarely cause lung infection, and most importantly, it is a more foodborne pathogen. Moreover, Bacillus anthracis is said to cause inhalation anthrax, and all other symptoms of this particular disease were similar to Mr. Rooney's that he have been felt before. So it was mentioned that after opening a dubious letter with white powder, he started to develop these symptoms and died within a few days. So that white powder could be the spores of the Bacillus anthracis, which might have caused the inhalation of anthrax. I think that's all for the why Bacillus anthracis is the most logical answer for our final answer.
All right, thank you, Zulhakim. So I think we have reached to the end of the discussion. We all agree that the pathogen that cause the disease is Bacillus anthracis. So that's all. Thank you all.